In the cold vacuum of space, 250 miles above Earth's surface, an unprecedented microbial experiment has been running for over 2 decades. The International Space Station, continuously inhabited since November 2000, has become more than a human outpost. It's an evolutionary laboratory where bacteria are adapting to space conditions in ways that both alarm and fascinate scientists. Today, we'll explore how the unique environment of the ISS is accelerating bacterial evolution, creating potentially dangerous antimicrobial resistance, and, paradoxically, how these space superbugs might help us solve our antibiotic crisis on Earth. The International Space Station represents something unprecedented in scientific history, a completely closed microbial ecosystem that has been continuously inhabited for over 23 years. Unlike Earth's interconnected environments, the ISS is isolated, creating what scientists now term a closed system microbiome. This self-contained ecosystem forces microorganisms to primarily interact with each other, spacecraft surfaces, and the human inhabitants, creating evolutionary pressures unlike anything on our home planet. Dr. Sarah Wallace from NASA's Johnson Space Center describes the station as an accelerated microbial evolution laboratory, compressing what might take decades on Earth into just a few years in space. The isolation of this system has led to remarkable changes in the microbial community. Through sequential sampling over the station's lifetime, scientists have observed a gradual shift in dominant microbial populations, with hardy, stress-resistant species becoming increasingly prevalent. Whole genome sequencing has revealed over 4,000 microbial species in the ISS environment, with approximately 12% showing significant genetic divergence from their Earth-bound relatives. These changes aren't random. They primarily appear in genes associated with stress response, metabolic efficiency, and most concerning, antimicrobial resistance. Recent findings from NASA's Gene Lab and Microbial Tracking 3 experiments have revealed something deeply concerning to microbiologists worldwide. Bacteria in space are developing antimicrobial resistance at accelerated rates and through novel mechanisms rarely seen on Earth. In April 2023, researchers led by Dr. Erica Hartman from Northwestern University published findings that sent shockwaves through the scientific community. Staphylococcus species isolated from the ISS exhibit resistance to at least 5 classes of antibiotics, drugs that form the backbone of our medical defense system. Even more alarming was the identification of a completely novel resistance gene cluster that appears to have emerged specifically in response to the environmental stressors of microgravity and increased radiation exposure. These aren't just minor adaptations. They represent fundamental changes in how bacteria survive and resist our medical interventions. The implications extend beyond the space station itself. These highly resistant microbes could potentially return to Earth, introducing novel resistance mechanisms into our terrestrial microbial communities. A February 2024 study in the journal Microbiome identified viable bacteria with novel resistance patterns on the exterior surface of returning cargo containers, microbes that somehow survived the extreme conditions of atmospheric reentry. While these particular bacteria aren't immediately dangerous to human health, they represent potential reservoirs of resistance genes that could transfer to Earth-based pathogens. What makes the space environment such a powerful driver of bacterial evolution? Several key factors create unique selective pressures that accelerate adaptation. First, without gravity's constraints, bacterial cells form entirely different three-dimensional structures, fundamentally altering gene expression patterns. Cosmic radiation aboard the ISS introduces mutations at rates 4 to 8 times higher than Earth's baseline, according to measurements from station dosimeters. This radiation bombardment creates genetic variations at an unprecedented rate, giving bacteria more opportunities to develop advantageous adaptations. Dr. Kasturi Venkateswaran of NASA's Jet Propulsion Laboratory has documented another fascinating adaptation. Bacteria in space form significantly thicker biofilms, protective communities encased in a self-produced matrix. These space-formed biofilms show up to 300% increased resistance to common disinfectants and can even incorporate metals from the spacecraft infrastructure into their cellular defenses. Perhaps most concerning is the observed rate of horizontal gene transfer among different bacterial species aboard the station. This process, where bacteria share genetic material directly rather than through reproduction, occurs approximately 24 times more frequently in space than in terrestrial settings. This allows resistance mechanisms to spread rapidly across bacterial populations that would rarely interact on Earth, creating novel combinations of resistance genes. Microgravity fundamentally alters cellular fluid dynamics, changing how bacteria sense their surroundings and respond to stress. Research published in the journal npj Microgravity demonstrates that the fluid shear forces bacteria typically experience on Earth are absent in space, triggering what microbiologists call the spaceflight phenotype. This distinct bacterial behavior is characterized by increased virulence, enhanced biofilm formation, and upregulation of stress response genes. Dr. Luis Z. from the University of Colorado Boulder has identified that this spaceflight phenotype includes significant changes to bacterial membrane composition. In microgravity, cell membranes become more rigid and less permeable, providing an inherent defense against antimicrobial compounds that would normally penetrate the cell. His team's 2023 paper in Frontiers in Microbiology documented how these membrane adaptations alone conferred a 37% increase in antibiotic resistance, independent of any genetic resistance mechanisms. This represents a completely different approach to antimicrobial resistance than what we typically observe on Earth. Rather than evolving specific enzymes to break down antibiotics, space bacteria are fundamentally restructuring their cellular architecture to prevent drugs from entering in the first place. This physical adaptation strategy proves remarkably effective and difficult to counter with traditional antibiotic approaches. Complicating our understanding of space-evolved bacteria is the discovery of what researchers term cryptic resistance. These are genetic adaptations that don't manifest as resistance under standard Earth-based testing conditions but emerge when bacteria experience specific environmental triggers. This means traditional antimicrobial susceptibility testing might completely fail to detect potential resistance in returning space microbes. The risk assessment model developed by Dr. Petra Schwender at the University of Edinburgh suggests that while the probability of a space-evolved superbug causing widespread problems on Earth remains low, the potential consequences could be significant. Her team's mathematical modeling indicates that even a single horizontal gene transfer event between a returning space-adapted microbe and an Earth pathogen could introduce resistance mechanisms our medical systems are unprepared to address. This concern has prompted space agencies to take action. The European Space Agency and NASA have jointly established the Planetary Protection and Microbial Return Working Group, developing new protocols for screening and containing biological material returning from space stations. These include advanced genomic surveillance techniques that identify novel resistance patterns before they enter Earth's microbiome. The standard decontamination protocols for returning astronauts and equipment were originally designed to prevent outward contamination of space, not to address evolved pathogens returning to Earth, a gap that now requires urgent attention as we continue our space exploration efforts. Surprisingly, these concerning findings have led to beneficial applications in terrestrial medicine. By studying how bacteria adapt to the extreme environment of space, researchers are gaining unprecedented insights into fundamental mechanisms of microbial adaptation. Dr. Lisa Knipp of the Massachusetts Institute of Technology has leveraged the ISS Bacterial genomic data to identify previously unknown regulatory pathways that control resistance gene expression. Her team is developing novel compounds that can potentially disable these pathways, creating an entirely new class of antibiotic adjuvants. These compounds don't directly kill bacteria but render them susceptible to existing antibiotics again, potentially breathing new life into our current antibiotic arsenal. The accelerated evolution in space provides a unique laboratory for studying how pathogens might adapt to future conditions on Earth, potentially allowing medical researchers to stay ahead of emerging resistance patterns. The NASA Gene Lab database now contains over 300 complete genomes of space-adapted microorganisms, providing an unprecedented resource for antimicrobial research. This vast dataset has attracted attention from major pharmaceutical companies, including Merck and Novartis, who have established research partnerships with space agencies to exploit this new frontier. Their approach uses machine learning algorithms to analyze the genetic changes in space-evolved bacteria, identifying novel drug targets that would be difficult to discover through conventional research methods. Perhaps most promising is the development of biomimetic materials inspired by the adaptive properties of space bacteria. Engineers at the University of California, Berkeley, have created self-sterilizing surfaces that mimic the radiation resistance mechanisms of space-adapted microbes. These innovative materials could potentially revolutionize hospital surface design and significantly reduce healthcare-associated infections, one of our most pressing public health challenges. The field of astromicrobiology has emerged from these studies, bridging space science and medical microbiology in ways that were unimaginable just a decade ago. By understanding how bacteria adapt to the harshest environment humans have ever inhabited, we gain insights into microbial adaptation that would be impossible to obtain on Earth. These space-inspired innovations extend beyond antimicrobial applications. The unique three-dimensional structures formed by bacteria in microgravity have inspired new approaches to biofilm management in industrial settings. The metal-incorporating defense mechanisms observed in space bacteria have led to novel bioremediation techniques for environmental cleanup of heavy metal contamination. Even the communication methods used by bacteria in the closed ISS ecosystem have provided insights into microbial community dynamics that are reshaping our understanding of the human microbiome and its role in health. As humanity extends its presence in space, understanding and mitigating the risks of microbial adaptation will be crucial for both astronaut safety and protecting Earth's delicate microbial balance from novel threats born in the stars. Future long-duration missions to Mars and beyond will face even greater microbial adaptation challenges, with extended isolation periods and different radiation environments potentially creating even more divergent microbial threats. The journey to Mars presents unique concerns. A mission lasting several years with no possibility of emergency return means any microbial problems must be addressed in situ with limited resources. The radiation environment during deep space transit and on the Martian surface differs significantly from the ISS experience, potentially driving even more unusual adaptations. Establishing robust monitoring systems and adaptive countermeasures will be essential to humanity's space exploration infrastructure. NASA and other space agencies are already developing next-generation containment systems, real-time microbial monitoring technologies, and space-specific antimicrobial treatments that can address the unique challenges of long-duration spaceflight. These systems must balance effective microbial control with the practical limitations of spacecraft resources and the potential impacts on astronaut health. The story of space-evolved bacteria reminds us that even as we venture beyond our planet, we remain fundamentally biological beings, carrying the microscopic world that has shaped Earth's ecosystems for billions of years. Our journey to the stars is not taken alone but in constant companionship with the microbes that have always been part of the human story. This coevolution presents both challenges and opportunities as we expand our presence beyond Earth. The ISS microbial experiment represents both a warning and an opportunity, highlighting potential biosecurity concerns while opening new avenues for addressing the growing antimicrobial resistance crisis on Earth. As we contemplate permanent settlements on the Moon and Mars, we must consider how human-associated microbes will adapt to these environments over generations. Will beneficial microbes that support human health also evolve in potentially harmful ways? How will the microbiomes of space-born humans differ from those born on Earth? These questions extend beyond immediate medical concerns to the fundamental nature of human biology as a spacefaring species. The microbes that have co-evolved with us on Earth for millions of years will continue to adapt alongside us as we take our first tentative steps into the cosmos. The International Space Station has revealed itself to be far more than a platform for human exploration. It's a unique microbial laboratory showing us how life adapts to the extreme environment of space. The accelerated evolution of bacteria aboard the ISS presents a double-edged sword. Potential biosecurity risks from novel resistance mechanisms returning to Earth, balanced against unprecedented insights that could help solve our terrestrial antibiotic crisis. As we've seen, bacteria in space develop thicker biofilms, altered membrane structures, and entirely new resistance mechanisms at rates far exceeding what we observe on Earth. The spaceflight phenotype fundamentally changes how these microorganisms function, presenting both challenges for astronaut safety and opportunities for medical innovation. The emerging field of astromicrobiology bridges our exploration of space with our fight against antimicrobial resistance on Earth. From novel drug targets identified in space-evolved bacteria to biomimetic materials inspired by their adaptive strategies, our cosmic laboratory may provide solutions to some of our most pressing medical challenges. As humanity prepares for longer missions to Mars and beyond, the lessons learned from the ISS microbiome will be crucial for both protecting astronauts and safeguarding Earth from potentially dangerous returnees from space. Our journey to the stars is ultimately a biological one, and the microscopic companions that have shared our evolutionary history will continue to adapt alongside us as we venture into the final frontier. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.